So let me say a few words about metabarcoding data. And you can read a lot about this in the module documents, so I will try to keep this short. So what is metabarcoding? Why do we do this metabarcoding actually? And the answer to this is basically to find the composition of microbes in a community. So even if organisms are unknown and not previously seen, we can still say something about them from this metabarcoding. And this may actually be the most important sort of motivation for using metabarcoding. You can still study sort of unexplored environments with this technique. So what is metabarcoding versus doing a full metagenome sequencing that we will also look at in bin 310? And some advantage of metabarcoding is that we can now easily map into unknown terrain. And this comes from this up here, right? This is not so easy with full metagenome sequencing. You can of course do it, but you can't say too much about the unknown part. With this technique, we can at least say something. It's cheaper, and that means we can sequence more samples. Both the sequencing itself will be cheaper with metabarcoding, but also the analysis part is simpler, or cheaper in the sense that it don't require heavy computational resources and stuff. And all this means you can do more samples for the same cost, right? Yeah, and the data processing is simpler, right? Don't require the expertise, perhaps. Yeah, I'm not so sure if that's a big difference, but at least it doesn't require the high performance computing, which we will do in bin 310 as well, and which we have to do when we come to this full metagenome sequencing. There are, of course, some drawbacks as well, and one major one is the lower resolution. You cannot, for instance, distinguish between species as easily as you would like to with this. You can distinguish genera and higher up in the taxonomy, but at the species level you struggle a little bit here. And if you go below species, like strain level, that's hopeless with this technique. And you also get no information about genomes in general, because with metabarcoding, we simply sequence a piece of the genome, not the whole genome. That's the whole idea, which we will see now. So, if you remember this figure here from another lecture, we sample metagenomes, right, or microbial communities in all kinds of locations and even within ourselves. We do the sample preparation and so on, extract DNA and sequence and do bioinformatics. This applies to all kinds of metagenomics, you could say. For full metagenome sequencing, we dig very deep, but for metabarcoding, we instead covers wide. So we give higher priority to having many samples across many different locations or environments or treatments, if it's about diseases and stuff, rather than digging very deep into a few samples, which is typically what we do in this part of metagenomics that we will also do in bin 310, but this is what we focus on now, metabarcoding. So, sampling the microbiota, and notice I use the term microbiota here, not microbiome. And I do that because I want to sort of emphasize that this is no microbiome as in many genomes. This is only a sample of the sort of taxonomy of the microbial composition. So the microbiota contains an unknown number of unknown organisms in an unknown mixture. And this is the headache we have to struggle with, right? When we do metagenomics, we sample from an environment. We don't know which organisms we sampled, and how many they are, and how they are composed relative to each other. So all these are sort of unknown to us. The idea of metabarcoding is to do what we call a targeted sequencing of specific markers. Marker genes, or it doesn't have to be a gene actually. Some of these markers are not genes. So we call them just specific markers, and that is just a piece of the genomes that are shared between these organisms, right? That's the whole idea. And we refer to this as amplicon sequencing because we first amplify these markers and then we sequence these amplicons, the pieces that we amplified. Those are the ones, that's the DNA we are sequencing. We can imagine we have a composition here then, and I always draw microbes like these circles because this is sort of the genome of each bacteria now. They typically have a single chromosome, circular. That's the most, not all have that, but that's the most common. So that's why I draw them like this. And I give them different colors here because they are sort of different organisms, then different taxa, right? And all of them have this marker, right? That's the idea. That's the idea of metabarcoding. All these, regardless of which one is there, they all have this marker somewhere in their genomes. And then we use this PCR amplification to just copy out this marker from each genome, and we don't care about the rest of the genome, and then we sequence these pieces. And after sequencing, we have then some reads, and here are the reads colored by which bacteria they originate from, right? So we can imagine that you have originally a hat of many sort of balls in it with different colors, a huge hat, and then you sample, in this case, 20 reads from that hat, and then the idea is that the fraction of, for instance, red reads here to the total of reads should now be the same as the fraction of how many red genomes are there compared to the total number of genomes in this hat, right? So by seeing this distribution here, we get an idea of how much we have of each organism. First of all, we see which organisms are there, the colors, and how much do we have of each. So relative number of reads from each organism reflects their abundance, their genome abundance, you could say. Well, that's not really the case, as we will see in the next slide. I mentioned it already here now, that notice I draw one marker on each genome. This is not always the case. Some markers occur in multiple copies on each genome, and it's not the same copy number. And that, of course, makes this information here less sort of directly proportional to the actual genome abundances. Composition of a sample is the same as the organisms we find and their relative abundance. So in this case, with these reads here, we would say the composition is like this: 25% red, 40% blue, 10% yellow, 25% green. If you compute here, there are 20 reads, and we have like 5 out of 20 is red, right? That's 25%. So this is a composition. And of course, instead of colors here, we have bacterial names or whatever. But in this drawing, it's colors then. So that's the idea. Think of a big hat that you sample from. You don't know what's in the hat, and you get the reads. And from the reads, you see that these 5 are similar. They should be grouped together. These 10 here are similar. They should be grouped. They must come from the same origin, right? And so on. And from that we can infer this composition here. So the 16S ribosomal RNA gene, that's the typical marker that we use. It's found in all prokaryotes. It's conserved enough for us to recognize in each, regardless of what type of bacteria or archaea we are looking at, we will always recognize the 16S. Different enough to separate between genera at least, but not between species, at least not always. And it has little horizontal transfer. That's also important, right? Horizontal transfer means bacteria transfer DNA between them, right? And that could be a problem if one bacteria has gotten the 16S from some completely different bacteria. But this rarely happens, extremely rare. And if they got from another one, it's usually a very closely related one, which is then also having the same 16S. So 16S is a nice marker for doing metabarcoding. But it's not the only marker, I should mention that. There are plenty of others. But this is the most typical one and the one we will use. It's a gene, it's a non-coding gene, around 1500 bases long. And this is what I told you in the previous slide. It's found in from 1 to like 18 or something like that copies, depending on organisms. So it's not like all of them have one 16S. By far, most bacteria have more than one 16S. And some have 2, and some have 5, and some have 10. So this is a little bit of a headache then, right? And we will look a little bit into this in an exercise in one of the modules. The 16S gene also have what we call variable regions. Look at this picture here. It's from an old publication of ours. And along the x-axis here, we have position, not in the gene, but we have made a multiple alignment here of many 16S genes. Lots of them, thousands, tens of thousands of them. And then even if one single gene is like 1500 bases, you have to add some gaps, right, in a multiple alignment. That's why the multiple alignment is wider than 1500. So in case you think that's strange, it's more than 3000 here. The point is that on the y-axis here you have entropy, which is a measure of variability between the sequences. So now we look at many tens of thousands together, variants of 16S, and then we can see that along this, so you can imagine all the 16S sequences are strung out along this axis here. You see that there are parts of the 16S having more variability than others. So these mountains here indicate high variability, and there are typical 9 of these mountains along the 16S sequence. And in between there is little variation. Here is quite conserved, right? In the valleys. It means that almost all of them are the same as here. So in order to distinguish between the different taxa, we should look into these regions here. And if you wonder about these colored vertical lines, they are the informative sites, as we say. And the higher they are, the more informative they are. This is where you really can discriminate between different groups of bacteria and so on. Here lies the information, right? But it shows that we don't really need to look at the whole gene in order to separate. Some regions are more important than others. And what we typically do, at least when we do Illumina sequencing, we amplify a piece of the 16S. We don't amplify the whole. It's actually quite long, right? It's 1500 bases. It's not so easy to do PCR amplification of very long DNA. So a shorter piece is safer. So we typically sequence, for instance, only this part here. We copy out only this part here, V3, V4 region, very popular, and sequence it. Depending on sequencing technology, yes, because with, for instance, nanopore or PacBio sequencing, which is long read technologies, then you would typically sequence the whole. So this is all this amplifying a piece is only relevant for Illumina sequencing, but that's the most common anyway, right? And for the long read technology, you can even amplify the whole operon and sequence the whole operon, not only the 16S, because 16S gene is part of an operon with 23S gene and so on. So that's possible then. But with Illumina, only these shorter fragments, and that's what we focus on now. The V3, V4 region is popular. You can see the two mountains. This is V3 and this is V4. They are really informative parts. Or out here, which is also popular. So, amplicons. Step 1, PCR amplification of targeted region. So we need to have a PCR primer pair. And the primer pair will, the primers will then bind to each end of this targeted region so that the PCR amplification can work on that between the primers, right? It's always the piece of DNA between the primers which is amplified. So we can imagine we have the genome here, piece of the genome, and here is the 16S gene that we want to amplify, but not the whole gene. We design primers that will match here and here. And this means that we will now amplify this part here from this primer to this primer. This will be copied out of the genome. This is the one we will sequence later. That's the idea. Primer bias, what's that? Well, the problem is that even if this 16S or whatever marker we use is highly conserved, it's not like it's 100% conserved. So it's very difficult to find primers that will bind to absolutely all organisms here, right? They are not identical. Nowhere, even along the 16S, which is highly conserved. So no primer pair is universal. Even if some primers are called universal, they are not really universal. They are perhaps close to, like 90% or something, but not 100% universal. So some organisms will not amplify, or at least amplify very poorly, which means there may be some bacteria in your hat that you never sample from, right? You don't see them by this technology, simply because your primers doesn't pick them up. They don't bind as they should because their 16S is too different from what your primers expect. So here we have an example of two such primers, forward and reverse. And notice that inside here we have not only A, C, G, and T. For instance, here we have an R and a B here, and this you will, and an S over here. You will see this in primer sequences. And what do I mean? This is what we call the UPAC ambiguity symbols. And an R, for instance, that we saw here means either A or G. That's what this means. Either A or G. So it means that the forward primer here is not actually a single sequence. It's actually more than one sequence this, because in this position you can have either A or G. Here, there B means these three. And here S means these two. So this single sequence here actually represents 12 different sequences. And why is it 12? Because it's two possible choices here combined with 3 possible choices here, which make 2 times 3 is 6 possible choices, and 2 here as well. So 6 times 2 is 12. So this is actually 12 different sequences that we use as primer, forward primer. And that is in order to bind to more of the bacteria or archaea, right? So it's not one single forward primer, it's 12. And one of them needs to bind. So we create the sort of degrees of freedom in this way. And you can typically see the same for the reverse. Here you have a Y and a V. So remember this, whenever you see primers, look for these ambiguity symbols, and you will see that these are actually representing more than one sequence. All these sort of universal or almost universal primers are like this. Okay, amplicon sequencing principle. First of all, multiplexing is a phrase that you run across. What is this? It simply means sequencing many samples in one run, and this is done only for practical purposes. Because I should say a little bit first, why do we do this? Because the sequencing machine, once you start a run, it will produce a lot of reads. And it's a waste of resources to sequence each sample that deep with that many reads. We don't need that for this metabarcoding. So instead of sequencing one sample per run, you sequence many samples per run, just to save money or resources. And that means we have to sort of mark the amplicons from different samples, and that is done by what we call a barcode sequence. And this is simply a short DNA sequence we add to the primers, and we add it, right? This is a technical thing that we add, and we know exactly which sequence has been, which barcode has been added to each sample. So it's unique to each sample. And here you can then see the amplicons. And notice all the amplicons have the primers. That's this lilac region here. But these samples, these amplicons comes from the first sample, and they have got a red barcode as part of their primer, right? While these are from a second sample, they have a blue barcode as part of the primer. And these one from a third sample with a green barcode. So it means that after we have sequenced, these barcodes are also sequenced, and then we can see from the reads from which sample they originated. And we can sort it into samples again. That's demultiplexing then. So step 2, sequencing of the amplicons. In here we have amplicons then from different samples, we can see, because of the different colors of the barcodes. And we do Illumina paired end sequencing. At least that's what we focus on here now. So let's consider one amplicon here. Then we do the R1 and R2 paired end sequencing like this. And notice it includes the barcode part and the primer part in both directions. So this is actually what we are interested in, the greenish part here. But we get all this technical part also. And now we can do demultiplexing, because the reads will carry with them this barcodes signal, right? And the primers are the same for all reads, regardless from where they come. So that's easy to recognize. But we are actually interested in this part here. And notice now the amplicons are typically so short that the reads overlap. The reads overlap each other. We get a part here which is sequenced twice. This is not strictly necessary, but this is what typically is done. So the fragment lengths, that's the length of the amplicons. We call it I. All of them are very similar, of course, because it depends on where the primers bind, and they bind more or less at the same positions relative to each other. That's how they are designed. So all the fragments are almost of the same length, so the reads will overlap almost the same amount each time. And we sequence by read length capital L. So we typically then, or what is typically done, we select primers such that this fragment length is shorter than 2 times L. So you get this overlap. This is, again, I have to say, this is not strictly necessary. I have often wondered, why don't we use longer fragments and just sequence each end of them? We could have done metabarcoding just as well with that. But I think that this has sort of been a standard from the beginning, and difficult to get people to do something new in this world. So it means we get these overlapping reads, and then we have to try to deal with that. So what do we do now? Read processing, and that's something you are going to do a lot of now. Well, first you do some filtering and trimming, some pre-processing, we say. Discarding reads with very poor quality. We can trim the 3' end, because we know that at least with Illumina sequencing, the 3' end is often the poorest quality. So instead of just throwing the whole read away, we can just take away some part of the ends to make where it is most error prone. Yeah, this is something we can do, or we may not do. Where this, to what extent we do this will depend on the quality of our data. And then we merge the read pairs, and that means we need to have read pairs overlapping, like we saw in the previous slide. Otherwise, this step will not be possible to do. But that's typical for Illumina data, that we have overlapping reads. So then we merge them. So we get from read pairs to one single read, which is longer, right? Cover the whole amplicon. The whole amplicon is now covered in one single read. So we glue together the forward and reverse, or R1, R2 read into one long read. And then we do dereplication. What does that mean? Because remember now, we sequenced these amplicons which are from these markers, so many of the reads will be identical to each other. And instead of storing 1,000 copies of the same thing, you store only one and say how many copies did I have of this one. Only the unique reads. That's dereplication. And then we have to do what we call, remove what we call chimera. And what is this? Chimera are actually artifacts of the PCR amplification step. And it results in reads which is a mixture of two templates. Let me show you this drawing here. So imagine you start out amplifying this blue gene here, or the 16S from a blue organism here, right? And you start copying it like this. And then for some reason, this copy process terminates before it should. It should have copied all the way out, but sometimes this doesn't happen in the PCR machine. It stops here for some technical reason, and then this piece here will float around in the PCR machine until the next cycle starts. And then this will be acting as a primer, because it's a shorter piece, right, that floats around together with the primers. And now imagine there is another bacteria, a green one, which is different from the blue, but they are identical up to here, right? So they differ only in the next part here now. And then this blue fragment from the first copying of this one will bind here, because it's similar enough. Doesn't have to be identical even, it's similar enough to this one to bind, and it acts like a primer, and then we start building the rest here. So this means you can end up with reads who are in one end copies of one organism, and at the other end copy of another organism. Mixture. And that's what we call chimeric sequence, or chimeras. And of course these ones we don't want. They are artifacts, right? Throw them out. So that's what chimera removal is all about, trying to find this and throw them out. Yeah, so it's also part of this step here. I have to say it's not that common to see this, or yeah, they are not overflowing the reads with the chimera. It's rather rare, I would say, but it happens. And especially if you go looking for sort of new type of bacteria, this will look like a new bacteria, right? So you can write a scientific publication, I found a new bacteria, but it's actually just an artifact, a mixture of two old ones. That's a disaster, right, if you do that. Okay, what more? Read processing. The clustering part is quite important, and typical of metabarcoding. So what does it mean? It means that reads coming from the same genome or organism or taxon, depending on the resolution we are looking at, they should be grouped. And each such group I now refer to as an OTU, Operational Taxonomic Unit. That's a standard phrase for each group, right? Each group is an OTU. So reads coming from the same origin, you could say, should be grouped. And there are two sort of very distinct approaches to this clustering. One we can call similarity clustering, which is sort of the historically the first way of approaching this. Computes identity between reads. I mean, a similarity as in how identical are the reads. And reads with a minimum identity are clustered. So if they are similar enough, they belong to the same group or OTU, right? And the idea is to always start at the most abundant read. Yeah, we will come to that. The most abundant read in the group is used as a representative sequence. We call it a centroid because each group should be represented by something, right? And then it's really it's natural to use the most abundant one as the representative. But it represents many slightly different sequences, right? Depending on how how strict you are in this criterion here. We can also do this in a different way, clustering by denoising. And what's the idea here? Well, we say that we group only sequences whose difference is due to sequencing errors. So we don't really look at similarity, at least not only at similarity, we will see. It relies on estimates of sequencing error rates. So we try to sort of denoise or take away the noise introduced by sequencing errors and only group those sequences who would have been identical if we hadn't had this sequencing error. And again, the most abundant read is the centroid. That's the same as up here. So one representative for each group, and that's the sort of the biggest one in the group. So this typically results then in a set of centroid sequences, OTU1, OTU2, blah, 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 and the sequence. You can see it's sort of a FASTA format here. It's typically a FASTA file. One sequence for each OTU, the representative sequence for each OTU. And then we take these sequences with us to each sample and we align them and count how often do we see these OTUs in each sample. And then this gives us then what we call the read counts. And the read counts we typically organize in a table like this, where we have one column for each sample and a row for each of these groups, OTUs, clusters, right? And the numbers here refer to now the number of reads. So this group are represented with 129 reads in the sample 1, but 257 reads in sample 2, and so on and so on. And the second group, 3,211 reads in sample 1, 1,973 in sample 2, and so on and so on. So this is straightforward, right? Read count table. How often do you see the OTUs in each sample? And now think back to the composition we saw with the colors. That's a column here now, right? What is the composition in sample 1? Yeah, that's how many you have of the different, each of these groups represents some bacteria or archaea. How much do you have of each of them in this sample? And how much do you have of each of them in this sample? And so on. That's the compositions. Similarity clustering. How do we actually do this similarity clustering? Well, Order all unique reads by abundance, largest first. And when I say all, I mean actually all. All reads from all samples are often merged together in one superset. But that's not really important. You can do it sample by sample as well. Use first read as centroid, and first means largest, right? The one with the highest copy numbers or read counts. The most common read. All other reads with dissimilarity less than D to this centroid belongs to the same OTU. So here comes a similarity, right? Or lack of similarity, dissimilarity. That's two sides of the same thing. Remove the clustered reads from the list and start at one again. So you just do this repeatedly over and over again until all reads have been clustered. So let me give you a visual example here. So here we have a number, each green blob here is a read, and the size of the blob is actually the abundance. So this is the most common read here, the second most and third most, and so on. The smaller the dots, the fewer we see of these reads after dereplication, right? So this one has many copies, not so many here, and this is the smallest one. Then we typically start here by the one with the most, and we draw sort of a radius around it, and that's the D, the dissimilarity, huh? So we say that, okay, everything within this radius of this one belongs to the same group as this one. So we color them the same color, for instance red in this example. And then these reads are now clustered and set aside. And then we go back to the ones who are not yet clustered, and we choose this, which one is the biggest one here now. It's only these two left, and this is the biggest one. We make another radius around it, right? And notice now this one here could have been member of this group, but it has already been assigned to the red group, so it's out of the assignment now. So this one will only group these two now. And this is what we call a greedy algorithm, because you start at one end and you never go back and change. So because this one was assigned to the red cluster in the first round, it cannot be assigned to the blue in the second round. You don't go back and change things. That's a greedy algorithm. You just continue, 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 and never look back. So this is a greedy algorithm, but it works pretty well. Pretty well. Simple. And notice it only depends on the dissimilarity, the radius, right? So what's the denoising? And let's look at what's called the data denoising algorithm. We will not dig into it. We will just briefly try to understand the idea of it. This can be sketched like this. We will use the Data2 software in bin 310, so that's why I focus on this algorithm here now. Start by all sequences in the same cluster. So that's sort of the opposite of what we saw in this previous slide, where everything was sort of scattered around and we started to form clusters from one end, you could say. Here we start by every read is part of one huge cluster. But again, we select the most abundant one as the centroid. Then we estimate error probabilities from that given clustering, and we will not dig into how this is done here now, but we have to compare sequences and so on, right? To see how often do the different errors occur, or variabilities, right? And from these error probabilities, we can compute what we call an abundance p-value for each unique read, and that we will see shortly. If at least one of these p-values is too small, so there is a threshold here on this p-value. If there is one p-value which is too small, it's an indication that this cluster should actually be split into two clusters. So that means we now make a new cluster using the smallest p-value as the centroid in that new cluster, keeping the old clusters as old centroids as they were. So in the first round you will have going from 1 to 2 centroids. In the second round you go from 2 to 3 centroids, and so on. You split and split and split and split until and each sequence is of course assigned to its most likely centroid. And then you proceed here until all these abundance p-values are large enough. There should be no p-values left which is smaller than some threshold that we set from the beginning. So remember that in this similarity clustering in the previous slide, we had to sort of set this the radius, the d, right? Here we have to set the p-value, p, instead. So how do we estimate these errors and these p-values? Well, first we try to find what we call a substitution matrix. And here's an example of a substitution matrix. Notice we have the 4 bases as rows and columns. And inside here we have probabilities. And notice the sum of probabilities in every row is 1. And this we should read as follow. If one sequence has an A, if the mother sequence or the centroid sequence has an A, this is the probability that all the members also has an A in the same position. Right? And that's typically a high probability, because if the sequences belong to the same cluster, they should be similar to each other, right? So if the centroid has an A, the other members also has an A. That's the most, but some will deviate, right? There are small probabilities of deviating. And this is the same, and if it has a C, this is the probabilities of the 4 bases, right? If the centroid has these characters, these are the probabilities of the members having these characters. So example, centroid sequence or mother sequence, as they like to call it in the Dada2 language. Let's assume it's this sequence. Very short one though, but for example it works. And here's a potential child or member of the same group. And we can see it's quite similar, but it differs here. And it differs here. Otherwise it's identical. So in order to find the probability of having the child given the mother, we have to go and look up these probabilities here. So if the mother has A, the child has A, yeah, that's going here, right? That's 0.95. Multiplied with, if the mother is C, the child is A. Okay, then we have C to A. And that's very small. So now we get 0.95 times 0.01. And the probability drops a lot. And then we have identical, so we have all these high values multiplied. It doesn't change very much, close to 1. But here we have a small one again, going from G to A. G to A, that's this one. So you can see that the probabilities for this sequence given the mother is simply looking up into this table and multiply together the probabilities you visit when you go through the sequence like this. The abundance p-value is computed as follows. This mother or centroid cluster has capital N members already. This potential child is observed small n times. That's the copy number of this read, right? So this is the sum of all the copy numbers of all the member reads already there. So this is a big number. This is not so big. And the p-value is then the sum of binomial probabilities with this as a parameter from n, from the small n to the large n. Yeah. We can dig a little bit into this if you like in in our Thursday or Friday sessions. But that's what I will tell you about it from now. For now, if this p-value is large, then we can see that this. Then we can say that this read here, C, can very well be a child of M and should belong to this cluster. If the p-value is small, it cannot be a child of this M anymore. So here is where we sort of make a decision of whether this read here should belong to this cluster or not, depending on this abundance p-value. Let me show you an example again, which is sort of similar to what we saw with the identity or similarity clustering. So here we have some reads again, and the size of the blobs are actually the copy number of the reads, right? And again, the distances you see here reflects the similarity, right, between them. So if we did this similarity clustering, we would start here at this one, the most abundant one, make a radius around it, and group everything that falls within that radius. Let me now show how this abundance p-value clustering will deviate. Because the abundance p-value doesn't only depend on how similar they are, it also depends on the abundance of these potential children. All these around the big one here is now potential children of this mother here. And abundance p-values become large if distance is small enough and the size is small enough. That follows from the previous slide actually. That if this is the mother, a child has to be close to the mother, but it can't be very big. Because if it's too big, it's another mother. It's not a child, right? So with the data clustering, you can, for instance, get a result looking like this. The red group here, with this as a mother, will include these as children, but not this one. Even if it's very close, it is too big. It's not possible that you can get so many reads coming from this one just by error, right? Because it occurs too frequently. This must be another mother who is happened to be very similar to this mother, but it's not a child of this mother. Can you see that? So this will actually then end up as, and this one as a separate group. So that's the big difference you can see between these two approaches. That you can have OTUs now quite similar to each other if they are of almost the same size. And this was never a consideration in the similarity clustering. Then we only looked at the difference, and if you are close enough, you are a member, regardless of how big you are. Okay, taxonomic assignments is something we also have to say a little bit about here. For the entire data set, we now have the centroid sequence, and we have the read count table telling us how much we see of each OTU in each sample. And then we typically assign taxonomy to the centroids. This is not something we have to do, but it's often done. And how is it done? Well, we take the centroid sequences, look up in some database of known sequences, in this case 16S then, with some known taxonomy, and see if they are similar. And how do we do the lookup? This is based on various approaches that we can come back to later, or in other modules of BIN 310, K-mer or alignment based. The database you use is actually more important than how you do the lookup. And that's perhaps easy to understand, that if your database contains sequences which is similar to the ones you have just discovered or sequenced, then you will get some taxonomy. If they don't contain these type of sequences, you will not get the taxonomy. So that's pretty obvious, right? So the database you use here is very important. But this is the idea, right? You take your sequenced reads and see, have I seen something like this before? And if I have, and I know the taxonomy, I assume that this read is also the same taxonomy, right? Yeah. And some OTUs may be from new organisms and not get any taxonomy regardless of what database you have, right? Because we have never seen it before. You can't recognize something that you haven't seen before. So we get no taxonomy for these, but they are still important. And I would say that if you think back to metabarcoding, why do we use it? This is actually quite important. You use metabarcoding if you probe into new terrain, you could say. And then you must expect that this taxonomy is not found for all of them. Many of these will be new organisms. So that's why I think that this taxonomy assignment step is not that important for metabarcoding, because we must expect this to be many of these OTUs are still interesting, right? We have no name for them, but we know that they are there. So that's why this clustering is the natural thing to do and not the taxonomy assignment as much. So that's the metabarcoding in a nutshell.